A few years ago, with my colleague Emmanuel Charpentier, I invented a new technology for editing genomes. It's called CRISPR-Cas9. The CRISPR technology allows scientists to make changes to the DNA in cells that could allow us to cure genetic disease. You might be interested to know that the CRISPR technology came about through a basic research project that was aimed at discovering how bacteria fight viral infections. Bacteria have to deal with viruses in their environment, and we can think about a viral infection like a ticking time bomb. A bacterium has only a few minutes to defuse the bomb before it gets destroyed. So many bacteria have in their cells an adaptive immune system called CRISPR that allows them to detect viral DNA and destroy it. Part of the CRISPR system is a protein called Cas9 that's able to seek out and cut and eventually degrade viral DNA in a specific way. And it was through our research to understand the activity of this protein Cas9 that we realized that we could harness its function as a genetic engineering technology, a way for scientists to delete or insert specific bits of DNA into cells with incredible precision that would offer opportunities to do things that really haven't been possible in the past. The CRISPR technology has already been used to change the DNA in the cells of mice and monkeys, other organisms as well. Chinese scientists showed recently that they could even use the CRISPR technology to change genes in human embryos. And scientists in Philadelphia showed they could use CRISPR to remove the DNA of an integrated HIV virus from infected human cells. The opportunity to do this kind of genome editing also raises various ethical issues that we have to consider, because this technology can be employed not only in adult cells, but also in the embryos of organisms, including our own species. And so together with my colleagues, I've called for a global conversation about the technology that I co-invented so that we can consider all of the ethical and societal implications of a technology like this. What I want to...
Let's fast forward just 15 years from now. Let's pretend it's the year 2030, and you're a parent. You have your daughter, Marion, next to you. And in 2030, she is what we call a natural, because she has no genetic modifications. And because you and your partner consciously made that decision, many in your social circle, they kind of look down on you. They think you're like a Luddite or a technophobe. Marion's best friend, Jenna, who lives right next door, is a very different story. She was born a genetically modified designer baby with numerous upgrades. Yeah, upgrades. And these enhancements were introduced using a new genetic modification technology that goes by the funny name CRISPR. You know, like something's crisp. This is CRISPR. The scientists that Jenna's parents hired to do this for several $1 million introduced CRISPR into a whole panel of human embryos. And then they used genetic testing, and they predicted that that little tiny embryo, Jenna's embryo, would be the best of the bunch. And now, Jenna is an actual real person. She's sitting on the carpet in your living room, playing with your daughter, Marion. And your families have known each other for years now. And it's become very clear to you that Jenna is extraordinary. She's incredibly intelligent. If you're honest with yourself, she's smarter than you. And she's 5 years old. She's beautiful, tall, athletic, and the list goes on and on. And in fact, there's a whole new generation of these GM kids like Jenna. And so far, it looks like they're healthier than their parents' generation, than your generation. And they have lower healthcare costs. They're immune to a host of health conditions, including HIV/AIDS and genetic diseases. It all sounds so great. But you can't help but have this sort of unsettling feeling, a gut feeling, that there's something just not quite right about Jenna. And you've had the same feeling about other GM kids that you've met. You were also reading in the newspaper earlier this week that a study of these children who were born as designer babies indicates they may have some issues like increased aggressiveness and narcissism. But more immediately on your mind is some news that you just got from Jenna's family. She's so smart. She's now going to be going to a special school, a different school than your daughter, Marion. And this has kind of thrown your family into a disarray. Marion's been crying, and last night, when you took her to bed to kiss her goodnight, she said, "Daddy, will Jenna even be my friend anymore?" So now, as I've been telling you this Imagine 2030 story, I have a feeling that I may have put some of you into this like sci-fi frame of reference, right? You think you're reading a sci-fi book, or maybe like in Halloween, mode of thinking. But this is really a possible reality for us just 15 years from now. I'm a stem cell and genetics researcher, and I can see this new CRISPR technology and its potential impact. And we may find ourselves in that reality. And a lot will depend on what we decide to do today. And if you're still kind of thinking in sci-fi mode, consider that the world of science had a huge shock earlier this year, and the public largely doesn't even know about it. Researchers in China just a few months ago reported the creation of genetically modified human embryos. This was the first time in history. And they did it using this new CRISPR.
As a test, they engineered 2 mosquitoes to carry the anti-malaria gene and also this new tool, a gene drive, which I'll explain in a minute. Finally, they set it up so that any mosquitoes that inherited the anti-malaria gene wouldn't have the usual white eyes, but would instead have red eyes. That was pretty much just for convenience, so they could tell at a glance which was which. So they took their 2 anti-malarial red-eyed mosquitoes, put them in a box with 30 ordinary white-eyed ones, and let them breed. In 2 generations, those had produced 3,800 grandchildren. That is not the surprising part. This is the surprising part. Given that you started with just 2 red-eyed mosquitoes and 30 white-eyed ones, you'd expect mostly white-eyed descendants. Instead, when James opened the box, all 3,800 mosquitoes had red eyes. When I asked Ethan Beer about this moment, he became so excited that he was literally shouting into the phone. That's because getting only red-eyed mosquitoes violates a rule that is the absolute cornerstone of biology, Mendelian genetics. I'll keep this quick, but Mendelian genetics says that when a male and female mate, their baby inherits half of its DNA from each parent. So if our original mosquito was AA and our new mosquito was AB, where B is the anti-malaria gene, the baby should come out in 4 permutations: AA, AB, AA, BA. Instead, with the new gene drive, they all came out AB. Biologically, that shouldn't even be possible. So what happened? The first thing that happened was the arrival of a gene editing tool known as CRISPR in 2012. Many of you have probably heard about CRISPR, so I'll just say briefly that CRISPR is a tool that allows researchers to edit genes very precisely and easily and quickly. It does this by harnessing a mechanism that already existed in bacteria. Basically, there's a protein that acts like a scissors and cuts the DNA, and there's an RNA molecule that directs the scissors to any point on the genome you want. The result is basically a word processor for genes. You can take an entire gene out, put one in, or even edit just a single letter within a gene. And you can do it in nearly any species. Okay, remember how I said that gene drives originally had 2 problems? The first was that it was hard to engineer a mosquito to be malaria resistant. That's basically gone now, thanks to CRISPR. But the other problem was logistical. How do you get your trait to spread? This is where it gets clever. A couple years ago, a biologist at Harvard named Kevin Esvelt wondered what would happen if you made it so that CRISPR inserted not only your new gene, but also the machinery that does the cutting and pasting. In other words, what if CRISPR also copied and pasted itself? You'd end up with a perpetual motion machine for gene editing. And that's exactly what happened. This CRISPR gene drive that Esvelt created not only guarantees that a trait will get passed on, but if it's used in the germline cells, it will automatically copy and paste your new gene into both chromosomes of every single individual. It's like a global search and replace, or in science terms, it makes a heterozygous trait homozygous. So, what does this mean? For one thing, it means that we have a very powerful, but also somewhat alarming new tool.
So, genome-engineered humans are not with us yet, but this is no longer science fiction. Genome-engineered animals and plants are happening right now. And this puts in front of all of us a huge responsibility to consider carefully both the unintended consequences as well as the intended impacts of a scientific breakthrough. Thank you.